video, we looked at some monohybrid crosses with complete dominance. Now we are going to look at incomplete dominance and codominance in these following videos, and then what their genetic crosses will look like. So what is incomplete dominance? None of the two alleles of a gene is dominant over the other, resulting in an intermediate phenotype in the heterozygous condition. So basically what we are doing is we are creating something new. And then we'll look at codominance. Both of these are extremely interesting. Like I said in the first video, genetics itself is very interesting. And now we're stepping into the area of where breeding comes in with animals, how farmers know what kind of cow will produce a lot of milk, and then they try and reproduce that same milk production in our offspring. The good-looking cattle, for example, this is also where game breeding comes in. All these varieties of wildebeest, the golden wildebeest, you've got black impala a few years ago that was very big on the market. So all of this is genetic. So these guys that are working with this have to be very clever in order to get what they want out of their offspring. So incomplete dominance, this is a question out of an old exam paper. So just so you guys know where it is from, let's look at it. So two snapdragons are crossed. What are snapdragons? These flowers over here, they are flowers, they are snapdragons. So one is homozygous white, so that one over there, and then the other is homozygous red. All offspring are pink. Now, how on earth does that happen if the parents are white and red, and then they're homozygous white, which means they look like that, and homozygous red, which means they look like that? So how on earth do you get to pink? And then they want to know, use a genetic cross to show these results. How did we get to pink? So this is where incomplete dominance comes in. If we look at the definition again, none of the two alleles of a gene is dominant over the other, resulting in an intermediate phenotype. Okay, so as I told you in the previous video, if something is dominant, it is written with a capital letter, and if something is recessive, it is written with a lowercase. Now in this case, both of these parents are homozygous dominant. They both have capital letters. So how do we get to the pink form? This is where the genetic cross now comes in. If you're unsure when it is incomplete dominance, when it is codominance, or when it is complete dominance, because they're not always going to tell you in the exam you are now working with incomplete dominance, you need to be able to draw the info out of the question that is asked. So in this case, you see that there are two parents, and neither one of them is dominant over the other. If that was the case, then one of them would have been a capital letter and the other one a lowercase. And then you've got an intermediate form in the offspring that results. So this is already telling me that it is incomplete dominance. That is why it is extremely vital for you to know your terminology. So let's do this genetic cross and see what comes out. So remember your format. We are dealing with the P1 generation, the first generation of parents, and we are going to start with the phenotype. So the phenotype is homozygous white crossing with, just want to get rid of those things on the side, so I've got lots of drawing space, crossing with homozygous red. Then the genotype, what would that look like? The genotype would then be capital letter W, crossed with capital letter R, capital letter R. Please make sure, because it has happened in previous papers where on previous years where the children, their capital letters aren't very clear. So if you're a cursive writer, make sure your letters are nice and clear as well. So make sure that your W is nice and pronounced. For example, if you were using a heterozygous, make sure that your W's are nice and pronounced, because it's not like the R where it's clear and definite which one is the dominant one. Just so you guys know. Okay, so now we've done the phenotype, the genotype. Now we're going into meiosis so that we can get the gametes that are formed. And then you are going to do your Punnett square. So capital letter W, capital letter W. What have I forgotten? Forgotten to write my gametes. Sorry. Remember to separate them so that we know that the alleles are separated. You can draw them in little boxes like this. Some learners have done it in the past when I was marking last year. They also wrote it like this, and we did give them a mark. So just as long as we can see a clear separation for the gametes, that we know that your alleles are separated after meiosis. What happens after meiosis? Fertilization. So this is now the Punnett square where it comes in. Then you've got a capital letter R and a capital letter R. Remember, this column goes down for this W. So you can write it like that, or you can write it like this. This W also going down. And then these R's have to go across. Okay, remember, with incomplete dominance, the one character or the one allele is not dominant over the other, so they are not overshadowing each other, and then they cause an intermediate form, which would be this over here. What do we have to do now? We have to do P1 has been done, so F1 genotype. Start off with the genotype. When you get to F1, it's just easier to write down. So in this case, it's nice and easy. There are 4 RWs, 1, 2, 3, 4. Then phenotype, you could also say 4 pink, because we know that is the intermediate form. They told us there at the top that that is the intermediate form. So you could then also say 100% pink. Okay. They also told you that. So that is incomplete dominance. I don't think I put another one example in there. So now we can carry on, and we can now look at codominance. So codominance, let's look at the definition. So codominance, both alleles of a gene are equally dominant, whereby both alleles express themselves in the phenotype in the heterozygous condition. So whereas incomplete dominance, there was an intermediate phenotype, with codominance, both those alleles express in the phenotype. So we see both of them. Now, I've given you some diagrams here already, just so you guys know what it looks like. So if you've got a white cow going mating with a red cow, you're going to have complete dominance in the sense that it's going to be a white cow with red spots on it, because both alleles are shown in the phenotype. Flowers also happens in certain flowers. You've perhaps got a white flower with pink crossing with a pink flower. Both alleles are equally dominant, and then they both show up in the phenotype. So what does a cross like this look like? Once again, here's an example. So a cross between a white-furred bull, so obviously this is homozygous, and a red-furred cow, obviously RR, so it's also homozygous. So all their offspring are heterozygotes, RW, displaying both white and red fur. Use a genetic cross to show this. So do you remember in the incomplete dominance one, they told you showing all offspring were pink. So we know that is an intermediate form. Whereas with this one, they tell you displaying both white and red fur. So you know it is incomplete dominance because both alleles are shown in the phenotype. So let's get into this genetic cross. So P1 phenotype, it's going to be homozygous white crossing with homozygous red. And then the genotype, what will that look like? It's going to look like, so it's very easy to confuse codominance with incomplete dominance. Once again, know your terminology so that when you read the question, you are able to pull and extract information out of it. Okay, what do we need to do? Go into meiosis, and meiosis then produces the gametes that we require, showing the alleles that are separated, and then fertilization can happen. So fertilization, you've got your Punnett square then, which you will draw, and then we've got those alleles. So once again, that going down, you don't need to show these arrows, it's just so you know what I'm doing in case you are a bit lost still. Remember, you can please ask me questions on WhatsApp, and then I'll do a video with just answering your questions. I'll also send some exam questions on the group, and then we'll do that together. And then if you have any questions, we'll discuss that on WhatsApp as well. And then that brings us to our F1 genotype. Remember, I like writing the genotype. It just makes it easier to see what the phenotype has to be, because with your F generation, you can get a mixture of things happening there. So seeing the genotype written out here just helps you structure your phenotype answer. So then it would be for, well, let's say for, what do I, sorry, for white and red fur. Offspring. Okay. There we go. So, and here's the visual of what we just did. Okay, now let's look at blood groups, because blood groups are also very important. They like asking them. Now, a very interesting thing with blood groups is that they show both complete and codominance. So I'm going to explain this to you now, why that is, how can it have two dominances. So with blood groups, you know that we've got blood group A, we've got blood group B, AB, and O. Now, the reason that blood is completely dominant is because blood group A and B overpower O. So O is the recessive here, and then A and B are the dominant blood groups. Now, that is what that is the complete dominance. Codominance comes in with blood group A, B. So in this case, neither one of these overpowers the other. They are both equally dominant, and both are seen in the phenotype. Okay, so that is why you get a blood group AB. Now, when you write blood groups, you, in a genetic cross, there's a certain way of writing it. So you make, it's almost like an I, and then you've got the blood group at the top. So for blood group A, you would write it like that. Remember, you still need to show two alleles when you're writing it. So blood group A looks like that. Blood group B would then look like that. And blood group AB would look like that. And then O is the special one because O is written as, just do that over, is written as a lowercase I, and that represents O. So this represents recessive. So whenever somebody is blood group O, that actually means that they've got a recessive trait, because O is a recessive trait to have, being blood group O. Now let's quickly look at a genetic cross. Why does it show complete and codominance? We just discussed that. So if there is blood group A involved with a blood group O, then there will be complete dominance. But if it is blood group A and B together, it is codominance, because both of those will be shown in the phenotype. So a heterozygous man for blood group A, so it has to look like that. If there was, if there would have been that, it would have been blood group AB, and they would have told you. So a man that is heterozygous for blood group A will look like that, and not like this, because then it would have been homozygous. Marries a woman who has blood group O. Now we know if something is blood group O, it has to be homozygous because O is recessive. So use a genetic cross to show the phenotypic ratio. So we're going to have to show the ratio at the end of this genetic cross. So let's jump into it. P1, the phenotype with blood groups, you can just say blood group A, because you're going to show them in the genotype that you know what you're talking about. Has married a woman or crosses with blood group. Don't abbreviate like I have, and just don't have space. Then the genotype, what does it look like? We've just written it at the top there. So that is what it would look like. Then what do we have to go through is meiosis to produce gametes where we can see the alleles that are separated. So remember to separate those nice and far from each other. And then fertilization is going to occur. I just want to grab a Punnett square to make it nice and easy. Remember, you guys still need to draw this in your test. Okay, there we go. So going down to the sides, So A is obviously dominant, and the I, which is blood group O, is recessive. So this side, this is what you'll have. Then you can show the F1 generation genotype. So the genotype is going to be 2 of those and 2 of these. Then the phenotype, so 2 blood group A and 2 blood group O. Remember, they ask for the phenotypic ratio, so it's going to be 2 to 2, or simplified, the ratio would be 1:1. So that is how you do a genetic cross for blood groups, and I will do some more info on blood groups in the next video.